Great. Can you see the full screen where you want to, are you able to see the full screen?
Yeah, we can see that.
Not the split view.
Right, it's perfect.
Okay, great. Hi, everyone. Thank you for the opportunity for me to introduce to you our research. The topic of my webinar today is on characterizing the cis-regulatory elements using single-cell epigenomics. This is a topic that I have worked on for all my professional life. Let's discuss what is a gene regulatory code. You know, every gene is driven by sequences that we call cis-regulatory elements that are responsible for the spatial temporal expression patterns of expression throughout development and in disease settings. These elements are clustered binding sites of transcription factors, which bind and recruit chromatin remodelers and transcription machinery to lead to transcriptional activation in a context-dependent manner. So that's a paradigm that we have been operating for decades. And we also know in recent days that in recent years, through numerous studies, that changes of such regulatory sequence, either in the germline or in somatic cells, can actually drive disease pathogenesis. But it is still poorly understood how this regulatory information is encoded in the DNA sequence. Our hope is that by understanding the gene regulatory code, we can eventually understand how non-coding risk variants of human disease can cause disease and how that information can then lead to development of therapeutics and treatment. So that's the overarching goal of my lab and also of the field. How do people study this problem? The conventional approach is to use transgenic reporter assays, such as shown on the slides. This is a perfect example, a textbook example of how enhancers element are studied in Drosophila embryos. This is a gene known as even skipped. It is expressed in 7 evenly spaced stripes during Drosophila embryogenesis. And each stripe corresponds to a set of enhancers, for example, here, stripe 2 enhancer, that are both sufficient and necessary for even skip 2's expression in that location. And studies from Michael Levine and many others have taught us that the enhancer consists of multiple binding sites of transcription activators and repressors. And the combined action of these transcription regulators determine the activity of this individual element in space and in time. And you should also recognize that in different enhancers, for example, stripe 4, stripe 6, different combinations of transcription factors are responsible for their specific position and developmental timing. So that's the principle that had been learned through years or even decades of such careful dissection of individual elements. In more recent years, due to the advance of DNA sequencing technologies, massively parallel reporter assays, and the high throughput characterization of transcription factor binding has all been developed. And this gave us beginning more and more understanding of how individual elements act to control transcriptional activation. So all these studies have demonstrated that the grammar and syntax of regulatory elements are likely evolutionarily conserved, meaning that whatever sequence that are operating in even skip in this Drosophila embryo, if you take it, transfer to a different strain of Drosophila, a different species, it actually can also drive the same position in the same developmental stage, which indicate that this is a feature of regulatory code that they are widely shared across different animal species. There are limitations. One is this type of approach, one element at a time using transgenic assays. They're not easy to generalize to study all the regulatory elements. And they're limited so far in mammalian cells to functional assessment of regulatory elements in cell lines or a small number of cell types where we can isolate them and study them. They don't consider epigenetic mechanism and chromatin regulators that are responsible for cell type specific activities of the elements. And they're also ill-equipped to dissect the grammar and syntax of cis-regulatory elements in vertebrates, which typically involve large genomic segments that operate over long distance. To give you an example of what the problem is, try to understand the meaning of this paragraph. Every words, you can individually identify what they mean, but putting them all together, it's impossible to understand the meaning, in part because this is a randomly shuffled text. Just to give you the idea that if we just understand individual elements function, we're still far from understanding the meaning of the whole text. So that brings me to the new approach that my group and many others have taken. It's called epigenome profiling. So let's start with the idea of epigenome. Epigenome is a way to describe the covalent modifications to DNA, such as methylation of cytosine, or covalent modifications to histone proteins, such as acetylation or methylation of lysine residues on different histones. And it also refers to the three-dimensional chromatin structure and open chromatin and other aspects of the chromatin organization that all working together to ensure proper expression of genes. So we refer this to so-called epigenome because they're responsible for individual cells to memorize their identity and to transcribe the proper set of genes that makes neurons neuron and makes fibroblasts fibroblasts. And we can think about epigenome as the software operating system working on top of the hardware, which is the DNA. So epigenomes differ from cell to cell, and that makes different cell types distinct. And to study epigenome, over the last decade or so, researchers have developed a whole series of tools to investigate and determine locations of epigenetic changes we call epigenetic mark. So for example, using DNA's hypersensitive sites, followed by sequencing or a tag-seq, you can determine the open chromatin throughout the genome. And open chromatin typically correspond to transcription factor binding sites and actively engaged transcriptional regulatory elements. DNA methylation also can be mapped using whole genome bisulfite sequencing. And DNA methylation typically in human genomes are hypermethylated genome-wide, except for in locations where you see a dip of methylation levels that typically correspond again to regulatory elements, because at such locations, TAP enzymes are recruited to remove methylated Cs. You can also determine locations of various histone modification using a technique called ChIP-seq. For example, you can learn the promoters by mapping the histone modification H3K4 trimethylation, because this is where the enhancer promoters are located. You could also map the active enhancers by mapping where the histone K4 monomethylation or lysine 27 acylation located in the genome, because this is combination marks indicating actively engaged enhancers. And you can also map the three-dimensional structure using techniques such as Hi-C, which map long-range chromatin contacts through a technique called proximity ligation, followed by sequencing. So all these tools together give us a picture about gene regulation in each and every tissue and cell type. And using such information, we can then begin to decode regulatory information in the genome by using the latest deep learning tools. For example, a couple of years ago, David Kelly's group from Calico and Google developed a strategy to take large set of data, as shown on the left, generated through ENCODE, to train a deep learning model, which consists of convolution, a neural network, followed by a new layer called transformer layer, to essentially predict gene expression and epigenetic marks using sequence information alone. And the predictor does a very good job of making such predictions. So this really gave us hope that we actually can learn about gene regulatory information in the DNA sequence by performing large-scale data epigenome profiling, and then use the power of deep learning or artificial intelligence framework to understand what's inside each base. There are limitations, though. One is that the training data used to make this model is of variable quality. And that makes the performance of such models still inadequate. Secondly, most training data for such models come from cell lines or heterogeneous tissues. And that makes the performance of such models even worse, because you realize that cell lines and heterogeneous tissues are not the same as primary tissue types that carry out physiological functions. And current training scheme also treat every experiment tissue cell types as independent prediction test, which actually is clearly suboptimal, because let's say if I give you 10 different liver tissue data, they are related to each other. They're not independent. Therefore, parameters need to be optimized in such. So our approach is now to overcome this problem. We're now taking a new strategy. It's called single cell epigenome assays. If you can perform profiling of epigenome on an individual cell basis, we overcome the issue of tissue heterogeneity. We can reconstitute cell type specific epigenome maps and therefore determine the regulatory landscape of that cell type. So that depends on our technological advance. A few years ago, we adopted a strategy called single cell combinatorial indexing ATAC-seq, which was pioneered by Jay Shanduri's lab at University of Washington. So what this strategy allows us to do is to map open chromatin in individual cells, tens of thousands of cells in parallel. So I won't go into details of the molecular biology, but it suffices to say that at the end of the day, we can actually apply this techniques to many different tissues by also leveraging our automated pipeline. Another strategy we also invented is called single cell methyl Hi-C, and by name, you know, it's designed to jointly profile both DNA methylation and the 3D genome conformation. Again, I won't tell you the details, but it suffices to say that by mapping DNA methylation, thousands of cells in parallel, we can quickly sort out individual cell populations in the mixture based on their shared and cell type specific demethylation patterns. And then we can aggregate the high-C data from each cell population to reconstitute a pseudobulk high-C profiles for that cell type. And that allow us to then infer the chromatin domains and the long-range chromatin interactions in that cell type. And finally, we invented a tool known as paired tag, which allow us to simultaneously map locations of histone modification together with gene expression profiles from the same cell and hundreds or thousands cells, tens of thousands of cells in parallel. And again, this is a very much needed tool so that we can profile the covalent modifications to histone proteins genome-wide. So we can see from this multiple single cell multiomics approaches, we have the tool to really profile methylation of DNA, chemical modifications of histone proteins, as well as three-dimensional chromatin structures genome-wide. We have the ability to generate data, but one of the common challenges we all face when we deal with single cell genomic data sets is data sparsity. So that has been a bottleneck for all single cell analysis. But to overcome this bottleneck, my former student, Rongxin Fang, who is now a postdoctoral fellow at Harvard University, developed a tool known as single nuclear attack analysis pipeline for ataxia. The short name is SNAP-ATTACK. This strategy essentially achieves clustering of individual single cell ataxia profiles so that we can then perform subsequent analysis for each individual cell type. Compared to other single cell ataxia processing pipeline, the unique feature of SNAP-ATTACK is that it employed a powerful mathematical approach to achieve dimensional reduction. So let me explain what it is. It's a strategy known as spectral embedding. So let me explain. Let's say if each row here is a single cell ataxia profile. 1 indicates there is a hit there, 0 indicates there is no hit. If you have 100,000 cells, it's a lot of rows. And then you convert this matrix into a pairwise similarity matrix, where you can basically ask how each how every pair of cells are similar to each other. And you can use Jaccard index to evaluate that similarity. You can also use cosine function to evaluate that similarity. And once you have this pairwise similarity matrix, then you can perform a dimension reduction using standard dimension reduction techniques. And that basically allow you to generate these individual clusters. All right. So that has been quite a popular tool because it's achieved very high accuracy and a high performance. It's the kind of data sets that can be applied using SNAP-ATTACK is broad. It does not really perform really poorly on some data sets and well on other data sets. It's performed equally well. But there is a downside, which is memory usage and the speed of SNAP-ATTACK was lower compared to other approaches, in part because for every data set, you need to compute this pairwise similarity matrix. And when you get to 1 million cells, this can become very, very memory intensive. So my postdoctoral fellow, Kai Zhang, who is a spectacular computational biologist, thought hard and long, and he came up with a new strategy for doing spectral embedding that actually can be that essentially skip the step of generating the matrix, which is the most memory intensive and the speed slowest step. So basically, he came up with a new approach of SNAP. We call it step attack 2, which is fast and memory efficient and scalable, and even extensible. So the idea is this: what Kai did was introduce this more efficient spectral embedding algorithm that have a running time of linear running time relationship to number of nuclei, as instead of n square. So that vastly improved the memory efficiency and the speed. And when he compared the performance of this new SNAP Attack 2, achieves incredible performance in terms of accuracy, retain the same accuracy advantage as original SNAP Attack version, but it's achieved much better performance compared to all the other available single-cell taxi processing tools. And you can imagine that the same strategy can be applied not only to taxi, but also RNA-seq and pair-tag and Hi-C. And he has now shown that it's the same tool, SNAP Attack actually does a very good job for a single-cell RNA-seq, single-cell Hi-C, single-cell cut and tag, and so on. This is just a little bit more details. You can see the running time and memory usage, the comparison between another popular single-cell processing, a taxi processing tool called ArchR. So we basically, for pre-processing, SNAP Attack 2 certainly has advantage in time and memory usage. And also for dimension reduction tools, it also has much better performance shown here on this one in terms of memory usage compared to all the other tools. Okay, now I've just laid the background for both methodology, wet lab approach, and computation approach. So we are all set in terms of applying these latest tools to dissect epigenome profiles in complex tissues, right? So I would just want to share with you that we now have used this strategy to dissect dozens of tissues from adult human beings and construct the single cell atlas for major human cell types in the body. We also use these tools to dissect the human brain, the mouse brain, the human heart, and also the various tissues of mouse tissues across time and across different dimensions. So all our data is now available from this portal called Catalyst. It's a Catalyst basically where you can see individual cells data as well as cell type specific information. And you can also download all the data that we have produced. Some actually have not yet been published, but we are putting them there for you to explore and to download. There are there probably are now already millions of single cell taxi data in this portal from both human and mouse. All right, I will just share with you one published study, and then I'll tell you a bit about another unpublished study. So in this study, which we published a couple of years ago, we interrogated 30 adult human tissues using single-cell taxi approach. And we also combined our data with 15 fetal tissue single-cell taxi experiments performed in Jay Shanduri's lab. And we combined the result from both labs and generate essentially uniformly processed single-cell taxi data sets consisting of 1.3 million cells. And Kai performed clustering using SNAP Attack and identified 220 cell types, half of which are adult and half of which are fetal type. And from this, then we can discuss, we can actually construct cell type specific accessible chromatin maps, and this is what you can see from Catalyst. This is just a snapshot of a few epithelial type cells in the colon. And you can basically see that they are all separate and distinct from each other based on some of the marker genes that they define them. We called 1.2 million candidate regulatory elements using a common peak collars developed by Shirley Liu's lab called MAX. For each of the cell type, we called on average 100 to 200,000 peaks, and combined together all of 220 cell types, there are 1.2 million such elements identified. And we plot the accessibility using this heat map here, where each row corresponds to a regulatory element, and each column corresponds to one of 200 cell types. And you can basically have a general idea that these elements mostly exhibit cell type specific accessibilities, indicating their potential usage in that lineage. And the next question we ask is, can we use this information to interpret non-coding risk DNA variants? And we know that common strategies such as GWAS can review the genetic risk variants for various human diseases, such as cancer, heart disease, and so on. But such risk variants are not necessarily causal. And in fact, for each tag SNP, they are in association with dozens of other SNPs that are so-called in linkage disequilibrium. So they are not necessarily causal. However, if they are indeed causing transcription regulatory elements to change, the prediction is that the disease risk variants should be enriched in enhancers of disease-relevant cell types. And this can be assessed using a common statistical framework invented by your colleague Ben Neal and Alcus Price. This is known as stratified linkage score, this LD score regression analysis. I won't go into details of this as it's already published and widely used, but the outcome of this regression analysis is essentially identifying how likely a enhancer landscape from a particular cell type can explain the genetic heritability of a GWAS trait. And this is a result. Out of several thousand GWAS traits and diseases that we investigated, we were able to find strong and significant association between 240 traits and at least one of the 220 cell types that we investigated in this study. So the red color indicate a FDR value, and basically you can see that each column representing different traits and SNPs and disease, typically there is, for each trait and disease, there is a very limited number of cell types that are associated with them. For example, allergy asthma is strongly associated with just a handful of cell types, all immune cell in nature. These are CD4, CD8 T cell and adult natural killer T cells, immediately suggesting that the T cell regulation is a culprit of this disease. Type 2 diabetes is shown here to be associated with another set of cell types. They are mostly in the islet. These are beta cells, one or type 2 islet beta cells, or alpha 2 islet cells. So these cells, again, provide a picture that the misregulation of gene expression likely underscore the disease pathogenesis of this type 2 diabetes. Atrial fibrillation is associated strongly with gene regulatory landscape from the cardiomyocytes. Again, here you can see that adult and fetal atrial or ventricular cardiomyocytes all show such associations. So what is atrial fibrillation? It's a common condition presenting high risks to heart disease and heart failure. It's characterized by irregular heart beating. Shown on the left is a normal heartbeat, and on the right is atrial fibrillation, where you can see heart beating can be very irregular. And we show that using our maps, two of the fine mapped risk variants with high posterior probability is squarely on top of this open chromatin discovered in the ventricular or atrial cardiomyocytes. And this open chromatin or candidate regulatory elements, let's call them that way, are located in the intronic region of a gene known as KCNH2. What is KCNH2? It encodes a potassium channel protein. So what does potassium channel protein do here? It turns out that potassium channel protein is absolutely necessary for the conductance of action potential in the heart cells. Why? Heart cells beat because they are there's electric pulse provided by the nerve, innervating nerves, to the heart cells, and that induce a localized temporary depolarization. In normal heart cells, they have an active membrane potential, and basically this short pulse allows this membrane channels to open and causing an influx of sodium ions into the cell to make the cell membrane reverse its negative potential to a positive potential. But that is quickly followed by opening up another channel protein, another channel known as potassium ion. So potassium channel allows the efflux of potassium ions that basically quickly bring down the action potential from positive to negative. And you can imagine that without potassium channel protein, you will have a much longer time to balance out this action potential. And that is exactly what happens. If you delete, in our case, if we delete this element that harbor these two risk variants, what we see is a prolonged action potential. And that basically is because there is not enough potassium channel proteins produced in these cells. And this is what we learned from this study, where by mapping the open chromatin in a cell type by cell type throughout the body, we can now interpret non-coding risk variants, and we can learn how such risk variants cause or predispose individuals for human disease and traits, such as atrial fibrillation. And we have done this for 48 other disease and traits. We perform fine mapping. We intersect the fine mapped risk variants with the open chromatin in all the cell types and traits. What we end up is finding that out of 10,000 such highly likely risk variants, the 30% of them actually are directly overlapping with the candidate regulatory element in different cell types. And we can also predict what transcription factors they are affecting, what genes they might regulate. So we have now a table consisting of 3,000 risk variants that are likely causal. We can tell where they are likely disrupting the regulatory elements in the genome. We can tell you which cell type this element is active in. We can also let you know which transcription factor and what target genes they might affect. So this is a table that is available through this paper that you can already download and study. Now, let me shift gear a little bit to a more recent unpublished work. In this paper, essentially published in, I should say it's posted in a bioarchive. We interrogated the adult human brain, right? Obviously, brain occupies so much importance in our life, and it's deserves a lot of attention. So, in collaboration with Margaret Barron, Joe Ecker, and Ed Lang, we dissected 42 adult human brains. These are from 3 adult normal individuals whose death was caused by causes not related to cognitive function. And we generated profiles from 1.1 million cells. These are ataxia profiles using the same strategy as what we did last time in the previous work. The data was generated under the supervision of Sebastian Prezel, a former postdoctoral fellow who single-handedly built a team and also automated platform to carry out a study like this. And data analysis was carried out by a postdoctoral fellow, Yang Li, who is now on the job market. The brain samples are all collected under very, very rigorous quality standards. Basically, this is how the procedure for each brain samples and donors. All the necessary information was collected, pictures taken, and for each slice, it was carefully annotated and dissected. And this work is collectively performed by Dirk King from University of Washington, together with Ling from Allen Institute. And the samples that were dissected then was shipped to Salk Institute, where Margaret Barron's lab further dissect to individual brain regions. And then we did follow up using single cell ataxia pipeline that we have established at UCSD. So from 1.1 million cells profiled, we were able to cluster them into 107 cell types, corresponding mainly to glutamatergic neurons, GABAergic neurons. These are 2 major cell classes of the neuron types. One corresponds to the excitatory neuron type, one corresponds to the inhibitory neuron types. We also discovered multiple types of glial cells, namely microglia cells here, oligodendrocyte cell, which correspond to the myelin sheath making cells, oligodendrocyte precursor cells that are responsible for replenishing of oligodendrocyte when they are dead, and then other cell types such as astrocyte in the brain. And we can basically annotate each cell type based on the genes that mark our known marker genes. When the genes are highly expressed, you can basically see that the gene body is accessible as well as gene promoters. So this allows us to make inference about gene expression levels and then identify candidate regulatory, cell types. And basically what this shows is that for most of the cell types, they are present in the brain in a highly regional specific manner. So when you have the highest region specificity, that's what's shown here. And a lot of the inhibitory neurons here, they are mostly in cortex, and that's why they're lower in specificity. While a lot of the new glia cells, they lack specificity except for one type of basically astrocyte. We called, again, open chromatin in each cell types, identifying a total of 544,000 open chromatin by looking at all the 107 cell types. And their cell type specific accessibility patterns is shown here. Again, you can see that majority of them are showing cell type specific open chromatin. And then we perform the same kind of stratified LD score regression analysis to link individual cell types to disease such as schizophrenia and other traits. And we can identify that a large number of excitatory neurons and inhibitory neurons show very strong association with schizophrenia. Only 2 types of microglia cells show strong, significant association with Alzheimer's disease. This is consistent with previous studies that shows Alzheimer's disease is strongly linked to the misregulation of gene expression in these 2 microglia cell types. We can also link major repressive disorder to a few excitatory neuron types and bipolar disorder to a few different set of excitatory and inhibitory neurons. So we have this list for dozens of neurological disorders and traits. And one thing I want to emphasize is that when you think about neurological disease and traits, these are mostly genetics, and these are hard to understand what is the molecular and cellular basis for each disorder, because they are mainly diagnoses based on behavioral or cognitive deficiency. But molecularly, it's not known for most of them what is the cause and what's molecular pathways they are responsible. So our study here really provide a starting point for us to begin to interrogate the underlying molecular basis of each of these neurological traits and disorders. And we can provide more tools to assist that process. We now are able to link regulatory elements to their intended target genes by combining both RNA-seq and ATAC-seq data from the whole brain. And we can basically identify correlated elements that shows correlated accessibility with the transcription of nearby genes that allow us to call 265,000 pairs of enhancer promoter target relationships. And this is a couple of examples. Here is a gene known as BIN1, a major risk locus for the Alzheimer's disease. And you can basically see this is a risk variance for the Alzheimer's disease. It's squarely on this enhancer that was discovered in the microglia cells, and this enhancer is predicted to interact with BIN1. So we actually can use this to study how BIN1 in regulation is disrupted by the genetic variants. And now we have also adopted the transformer model, initially I described from David Kelly's group, to basically predict for each base in the genome how they are contributing. Can we actually, the end goal is, can we predict open chromatin using sequence information alone? Again, this is the same framework that was developed by David Kelly with some minor twist, which I won't go into detail due to time. But the idea is that now we actually have a very good predictor that can basically predict open chromatin from sequence information alone. This is, for example, a taxi from the at the BIN1 locus at the microglia. The yellow line is what we can predict, and the Pearson correlation coefficient is 0.74, which is not bad. And we can make such models for every major cell types, and for each of them, we can actually make really good predictors. The use of such predictor is shown here. We can perform in silico mutagenesis to identify key bases that are contributing to the predicted performance. And if, for example, if you change the base into one of A, C, T, Gs, you will see a degradation of that performance shown here as the blue color, and that typically corresponds to transcription factor binding motifs. So using this in silico mutagenesis, we actually can now begin to formulate which base and which transcription factors might be perturbed by the risk variants. So I won't go into details on how that is done, but I want to say this is a really exciting time because now we have big data. We also have the deep learning framework at hand to begin to understand how the neurological traits or how generally transcription patterns of genes can be encoded in the DNA. To summarize what I've just said, we are systematically investigating the open chromatin and epigenomes in human cells across the body. And this allows us to now develop sequence-based predictors of open chromatin and of epigenetic marks, and use that information, use that predictors, we are able now to systematically investigate the functional roles of risk variants. All the data I talked today are available at this Catalyst web portal. So feel free to explore and let me know what you, if you have questions. And I want to end by thanking my colleagues. I want to specifically mention Yang Li, who is shown here with open arm, a very talented computational biologist and who is on the job market. And I want to also mention UCSD Center for Epigenomics, a resource that I'm directing and which has been instrumental in producing the large-scale single-cell epigenome data, which I mentioned today. I also thank my collaborators, whose name I mentioned over the talk, and also the funding agency. I know we have a few minutes, and happy to take questions.
All right. Thank you so much for the great talk. Very informative. Do we have any question from the audience? If you have a question, please raise your hand or just type your question in the Q&A. We'll unmute you. Okay, there's two questions. All right, let me just unmute Michael Rogers first. All right, Michael, I think you can talk now.
Yeah, can you hear me?
Yes.
Okay, great. So I was intrigued by the transformer results that you were showing, and I recognize that you were leveraging that as a tool. Very often, biologists find themselves in the reverse position, or wanting to do the reverse of what you're describing there. In other words, we have cells or tissues that have where we see changes, but it is challenging for us to infer what upstream processes led to those expression changes. And I'm wondering where the process is, or where progress is in being able to infer, for example, different signaling pathways that have been activated to lead to the cell or the gene expression changes that one might observe in disease versus normal tissue, for example.
Michael, that's a great question. I don't have time to present that line of research, but we are also taking advantage of a recent work from Samantha Morris' lab called Cell Oracle. In this work, you can take single-cell ATAC-seq, single-cell RNA-seq combined together to make inference on gene regulatory networks. What this modeling approach allows us to do is to link gene expression to their upstream regulators. And that's, I think, is what you want. So,
so Cell Oracle is kind of the short answer as things stand right now.
That's right. That's what we are currently trying to. I don't have time to talk about that, but that's what we are doing.
Okay, thank you. I really appreciate it.
Sure.
We have a lot of questions from Wanqing. So Wanqing, I think you can unmute yourself.
Hi, so this is one. Yeah, so I have a question on the kind of like the final part, the prediction of the ATAC-seq signal. I was wondering, could you comment a little bit more on kind of like the subtle differences between the prediction data versus the true data, especially in terms of the signal strengths? And also, how should this be taken into consideration in terms of using the in silico mutagenesis?
Yeah, again, we are experimenting different type of framework. So deep learning is a rapidly evolving field, and you can do a lot of things. So we are getting 0.74 in terms of Pearson correlation. It's not perfect, meaning that a lot of peaks we are not capturing them, and we also are probably calling some false positives. We recognize that there's probably ways to improve it. One is probably by using a more optimal parameters in our deep learning strategy in training the model. Second is by including additional data sets, ideally from different species, so that we actually can filter out non-relevant information. So we are doing all of that, trying to actually improve this model prediction to as perfect as possible. It's not perfect, but it doesn't mean it's not useful. I think one of the utility I'm showing is potentially using this in silico mutagenesis to learn what transcription regulators might be actually involved in the process. So for some for one locus, for example, the BIN1, we can show that our in silico mutagenesis indeed correlate with risk variants, which we have studied in the past as causal, and make predictions on which transcription factors might be involved. In the future, I think we hope to be able to further sharpen our prediction, make this more and more reliable.
Yes, thank you so much.
Absolutely.
Great. Do we have any more questions? If not, maybe I can ask one question here. So when you actually talk about the predictions of the regulatory function of non-coding risk variance, you basically use the Bochner-seq data to get the sleeps, right? Just want to confirm that. Is that correct, Dr. Ren?
I'm sorry, I missed your question. Can you speak again?
My question is that, so when you actually talk about the regulatory function of the non-coding risk variance, is that the variance you identified by the bulk data, or is actually single sequencing data or single taxic data?
Yeah, let me clarify that. There have been thousands of GWAS studies that have been performed on human population. And if you look at the GWAS European Bioinformatic EBI database, they include 500,000 such GWAS variants. These are all genetic risk variants for various disorders. So we just take that.
Okay, so from the public database.
Yes, that's the data we start. So we then actually create so-called summary statistics for each trait and disease. Most of the GWAS studies lack the population sample size, make it not possible for us to actually have conduct specific power, with statistical power to do this association analysis. But for some of them, we can. So end of the day, we found 240 traits to be strongly associated with one or more cell types based upon the enrichment analysis that I described called LD score regression. So we don't have to do SNP detection because all the data we generated were from normal individuals. It's not from the patient, from normal individual.
I see, got it. Thank you. Well, I think there's no question in the Q&A. If we don't have any other question, thank you so much, Dr. Rin.
Thank you everyone for your attention, and hope to see you again. Bye-bye.